Hey everyone! So, throughout this podcast series, we've been talking about how life began and how it came to be as we know it today. It seems like life 2.3 billion years ago was another world, one where microorganisms ruled the world, but we're not far from that today, as we will see throughout this podcast. The astrophysicist Carl Sagan said that man tends to define in terms of the familiar, but the fundamental truths may not be familiar. This, of course, has many applications, and its meaning can be extended into different fields, but when you think about how little we humans have been on Earth, and how little we have recorded, and how limited our observations are, we can appreciate that what's familiar to us is nothing compared to what's familiar for a microbe. Microbes have experienced it all. They've been here for billions of years. Observing microbes today is an essential task for us to be able to understand where we came from, where we are, and where we can go, and just how far we can go. Microbes are a framework for our existence. So, my name is Lucia Belakova, and I'm here today with Dr. Daniela Jaramillo, the head of research for the Human Microbiome Project, or HMP for short. Thank you so much for being here, Daniela.
Oh, good afternoon, and you're welcome. Thank you for having me.
So, very broadly speaking, HMP is designed to map the microbial communities in our bodies and characterize them to be able to understand the microbial components of our bodies and how they contribute to our physiology and disease predispositions. As your colleague Rob Knight put it, microbes are doing things we never really suspected, right? Could you tell us more about that, though?
Of course. Well, we launched the Human Microbiome Project in 2007 in response to the unsuccessful attempts in the field of microbiology at identifying and culturing the organisms that make up the human microbiome. The premise upon which HMP was founded is that the humans are so-called supraorganisms, or a group of organisms which behaves in some respect like a singular organism. And as supraorganisms, we are composed of human and microbial components. HMP was designed in response to the ongoing technological and computational advancements of the Human Genome Project, and it's also a response to the massive transformation in human lifestyle that affects our health as a result of change in our microbial ecology.
Oh my God, that's a massive project, and I know because you told me this before, that you're collaborating with other human genome projects. So it's really one huge microbiological roadmap initiative. That is a huge step for humankind, and an exciting one too. Could you perhaps tell us more about this whole superorganism thing?
Sure, so prior to the results of the Human Genome Project, we thought that humans were comprised of around 100,000 genes. But it actually turns out we only contain 20,000 genes or so, pretty much the same as a fruit fly. It's very humbling.
Very humbling, yeah.
But if we consider ourselves to be a composite of our human genes and the genes of our microbiome, turns out that the number 100,000 is actually an underestimation. If we take all the genes into account, what emerges is a human supraorganism. If we really want to understand the range of our genetic and physiological diversity, we have to first characterize our microbiome and what influences the microbes' distribution and evolution. The outcome of this analysis would provide us with a perspective on how humans are evolving as of today and how our lifestyles affect our microbiome, which in turn affects our health and predisposition to diseases.
Wow, that's crazy. Okay, talk to me about the goals HMP set out to accomplish.
Well, the main goal of HMP is to characterize the human microbiome and develop a set of microbial genome sequences. So basically, we want to characterize our microbiome, or microbes, to know which ones are good and which ones are bad for us. From there, as I said it before, we set out to define the relationship between diseases and the human microbiome, or how the changes in the microbiome impact our health, or how we can go from the bad microbiomes to the good microbiomes. The task we have is to manipulate microbiota to optimize its functions, really.
Yeah, that's super fascinating. So what were the biggest challenges of the project, either at the beginning or even as the project progressed?
One of the biggest challenges we had when we started was the problem with scale. The technology for microbial DNA sequencing existed, but we're talking about 10 years ago, so remember that. And we had 250 microbial DNA samples from 250 healthy medicine students to sequence from 15 to 18 body parts, actually.
That's around like 4,500 samples, am I right?
That's right. Luckily, we've come a long way with metagenomics and specifically with the genome-anchored characterizations of the microbial communities.
Metagenomics, how does that work?
Yeah, well, that has to do with how we gather our data, or method, if you will. I'll tell you a little bit more about it. In the first place, we collected the samples from the 250 people from 15 parts of the body if they were males and 18 if they were females. Then we collected stool samples, saliva samples, vaginal samples, throat samples, tonsil samples, skin samples, and so on. You get the idea. Then we extracted the DNA from the microbes in those samples, amplified and sequenced the 16S rRNA or ribosomal RNA, then grouped the similar sequences into operational taxonomic units, and finally we took those into existing databases that allowed us to show the relative abundance in communities.
That sounds like a lot of work.
Definitely, but immensely rewarding work. Now we have these graphs, like the one you have in front of you, which shows just how vast the interpersonal and intrapersonal variability in bacterial lineage is. And do you know what this means? This means basically that the microbiotic composition differs greatly across the human body and also from person to person, even among healthy bodies, like the 250 20-something-year-olds we sampled.
Oh, yeah, you can very easily see that in this table right here in figure A. Like the occurrence of various microbes as well as the magnitude of the occurrences is way different depending on the person.
Yes, correct. Every vertical line you see across both charts is a single person.
Oh, I see. What's even more interesting from what I'm getting here is that the microbial composition is way different depending on the body area. Like the interior nares, or the outside creases of the nose, have a lot of actinobacteria, whereas the mouth comprises mostly of proteobacteria.
Yes. And what's even more interesting and obvious in the figures, without getting too technical about the bacteria names or anything, is that even though the microbial composition varies from person to person, they are still able to complete the same metabolic pathways as you can see in figure B.
Yeah, that pattern is evident, undeniable. You can see it right there. Figure A shows no relationship whatsoever between the samples, meaning they have completely different set of microbiomes. Each person has his or her own ecosystem of bacteria. But figure B shows that the functions are the same, and they're all in perfect health too. It's amazing. But this variation is among a very homogenic and, as you said, healthy people. I imagine unhealthy samples must not follow this trend.
Yes, exactly. You got it. Microbial communities from one person are as different to another person's as an ecosystem of a desert is to that of an ocean. And the functions in healthy people remain the same, but in unhealthy people they are not. You've got that right.
I've got to say, my mind's blown.
But wait, it gets even better. So my colleague Dr. Gordon and his team began investigating the role that gut microbes play in obesity. The team compared the intestinal microbiota in lean and obese individuals, and what they found was that the microbial composition of the lean people's gut was far more diverse than that of the obese individuals. And lean people, for example, have far wider variety of bacteroidetes, whose main function is to break down bigger plant starches and fibers into easily stored molecules that can later be used for energy. So yeah.
Yeah. Oh, yeah. I've heard about this. Was this the experiment based on baby mice that were genetically identical and were also raised in a germ-free environment so that their bodies contained no bacteria?
Yeah.
Yeah, and then they collected gut microbes of lean and obese people and planted these microbes in the intestines of the sterile mice. And one group of mice had the lean microbiota implanted in their gut, and the other group had the obese people's microbes. And they ate the same amount of the same stuff, but the mice had the obese microbes and their bodies still got fatter. And their microbiotic composition was obviously less diverse than that of the lean microbe mice.
Yes, that's exactly the one.
Yeah, I also heard that they conducted this experiment again, but they moved all the mice, the fat and the lean, into a shared cage, and this time they all remained fairly skinny. And what happened was that the mice carrying the obese people's microbes picked up the gut bacteria of the lean mice by consuming their feces, probably. But do you know what happened after?
Yeah, so then Dr. Gordon and his team transferred 54 varieties of bacteria from the lean mice to the obese mice, and all of them eventually lost weight. What Gordon concluded was that the obese mice were lacking microbes that helped the bodies maintain a healthy weight and metabolize faster. One of the missing bacteria was Helicobacter pylori, which regulates ghrelin, or the hunger-stimulating hormone. Funny enough, this hormone used to be very abundant in humans a long time ago, but the use of antibiotics under sterile living conditions have rendered it quite rare.
That's absolutely insane. So, is this a new way of treating obesity? Like, in the future, are we going to be able to just transplant microbes into our gut and lose weight? And can we do this with other diseases too?
Yes, potentially. And thanks to HMP, We are leaning towards a more probiotic approach to medicine rather than an antibiotic approach. And in the case of obesity in particular, researchers are looking into fecal transplants or enrichment of foods with beneficial bacteria strains, kind of like probiotic yogurts, but effective. And yes, we are looking into bacterial effects in all areas, from physiological treatment to microbe-based therapeutics to treat anxiety, anxiety and even maybe autism.
I'll definitely be calling you for a follow-up on that last part.
Yeah, I'll be happy to come back anytime, but I'll leave you with another thought regarding this. So fecal transplants have had an incredible success rate in curing diseases like C. diff, a case of severe diarrhea. And what's even more, the mice that we studied didn't only change the way they metabolized the food after the microbial transplants, but they actually changed the way they behaved towards the food. So, for example, mice with the microbes from the obese people started eating more. So microbes, these little organisms inside of us, are dictating the way we behave, what we do. We truly are superorganisms. It may be too soon to make any claims regarding this, but I just want you to consider how this changes things for medicine, our identities, our humanity. I mean, our human genes are 99.9% alike, but our microbial DNA is only around 10% alike. So really, if it were not for our microbiomes, we would not be able to tell each other apart. Genetically speaking, of course.
At the beginning, you mentioned how we didn't suspect microbes were doing all of these things, but now that I'm thinking about it, it makes perfect sense. Like, if you look at the most recent version of the Tree of Life from 2016, you can see that microbes, especially bacteria, make up most of the organisms that are alive today, and they have always outnumbered pretty much everything else. We're basically another host for them, another environment for them to exist in, and like you said, they outnumber our genome, our human cells, so we depend on them as much as, or even more, than we do on our own genetic information. It does make you wonder what makes us human, right?
Oh, sure thing. We live in a crazy world.
All right. Thank you so much for coming here, Daniela. Surely we'll keep in touch to discuss more about microbiology and perhaps some more philosophy as well.
Oh, my pleasure.